The broadcast is now starting. All attendees are in listen-only mode.
Well, good evening, everybody, and welcome to another edition of Question Lab. It's been a couple of weeks. It's been a minute, right, since we've held this. So we're glad that you have been able to reconvene as we tackle questions related to gastrointestinal. So we're going to do a bunch of questions on GI tonight and kind of help you learn how to attack these step one questions, as well as kind of focus on key content as it relates to GI. And we'll also have a raffle at the end of the night where some lucky person is going to walk off with a subscription to RX 360 Plus. So that's what we're going to do tonight. I am going to first introduce my co-presenter tonight, Sean Nanji. Sean, would you like to introduce yourself?
Sure thing, Jeff. My name is Sean. I've been working with USMLE-Rx now for a little over 3 years. Happy to help out tonight with our question lab. Been in medical education for a little over 10 years now. Looking forward to today's webinars, and glad you all are here.
We're going to tackle 4 questions tonight. And what you're going to see is that as we get into this, we're going to be reflecting our methodology, right? So what main thing for us is that we want you to be as prepared as possible on test day. And one way of doing well on any exam, but especially board exams, is to have a method. So we're going to start with question number 1, and what you're going to see first and foremost, there are no answer choices. And that is by design. So what we do is we hide those answer choices, and we recommend you hide those answer choices because we don't want an answer option to freak you out, or if you see something that it's like, oh gosh, I don't know what that is, or I can't remember what that is, or it's easy to kind of psych yourself out, especially when you're if you're anxious or you're in an area that you don't feel as confident in. So we say cover up those answer choices as step 1, and then step 2, we say read the lead-in first. So instead of starting with the vignette, which is the story, basically, right, the narrative behind the question, we ask you to read the lead-in. And in this case, the lead-in is, which of the following would be deficient as a result of the most likely gene deletion? Okay, so by reading the lead-in, now you have an understanding of what kind of information you're going to be looking for when we read the vignette, okay? And in some cases, there are the occasional questions in which you may not even need to read the vignette, right? So you want to be, again, we feel like this is a good way of being efficient as you tackle these questions. Okay, so you've read the lead-in. Now you can read the vignette. In a genetically modified mouse, researchers deleted a gene that is only expressed on the plasma membrane of enterocytes. When tested, the mice were found to have elevated high molecular weight proteins and complex lipids in their stool. So the question again, which of the following would be deficient as a result of the most likely gene deletion? What I'd like you to do is head down to the question box and let us know how many steps you think this is going to take in order to solve it. Okay, so you think this is a 1-step question, a 2-step, 3-step? Let us know, and I am going to pass the mic on to Sean. Sean?
Well, thank you very much, Jeff. So we're going to go ahead and show you what we believe are the most important clues in the vignette and lead-in. So as you can see here, they're giving us some good info here. So we need to figure out what's deficient as a result of the gene deletion. So we got a genetically modified mouse, deleted a gene. We know that it's only on the plasma membrane. And now we know that they have high molecular weight proteins and lipids in their stool. So we got to figure out, why it's there, right? Which enzyme or enzymes caused it, and which gene is responsible for this. This is likely a 2-step, maybe a 3-step question. So, that's kind of where we are now. So let's go ahead and take a look at these answer choices. And when we look at these answer choices, we like to start at E and work our way up. And the reason we do that is because we don't want you to, accidentally forget or overlook an answer choice. So this kind of forces you to go through all of the options, right? So let's start at E and work our way up. E, pepsin. D, lipase. C, enterokinase. B, chymotrypsinogen. And A, amylase. So once again, we've got these answer choices. We've got our lead in, our vignette, the clues highlighted. We will now give all of you a few moments here to lock in your answers as we open up that poll and let you lock them in.
Okay, excellent, Sean. So we've launched the poll. Go ahead and let us know which of the following you think would be deficient as a result of the most likely gene deletion. Kind of a tricky question. Get us started here. Maybe not necessarily what you would automatically think is a classic GI question, but this is the kind of question you're going to see, and you're going to need to be able to figure your way through it. Let's see. So we've gotten about almost half of you have voted so far. So let's give you a chance to remember. We always say this, be another reminder for you. You never want to leave a question unanswered on test day. You're not going to be penalized for wrong answers. Okay, so it looks like we've got a clear favorite here, and a couple that are kind of in the running, or at least in the running for runner-up. So I'm going to close the poll, and let's share these results. So over half of you went with enterokinase, with lipase and chymotrypsinogen garnering the second and third place votes. So interesting. Let's go ahead and see. We'll hide the results and share the answer. And the answer is indeed answer choice C. Great job, everybody. Good way to start off the evening. If you didn't get it right, that's okay. Sean's going to help break it down for you. Sean?
Well, thank you very much, Jeff. So the key to answering this question was realizing that we have proteins and fats, aka lipids, in the stool, right? So what's responsible for that? So what we need to do is figure out, what is on the plasma membrane of enterocytes and is responsible for activating trypsinogen into trypsin, because then the trypsin is going to activate other pancreatic enzymes like lipase, chymotrypsin, and carboxypeptidase. And these enzymes are essential in the breakdown of lipids and all proteins. And so it's going to be the gene for enterokinase, because enterokinase is expressed on the plasma membrane of enterocytes and is responsible for activating trypsinogen into trypsin. So although enterokinase is not a pancreatic enzyme, it is important for the subsequent activation of the proenzymes from the pancreatic exocrine organ. So, and once again, those pancreatic enzymes are lipase, chymotrypsin, and carboxypeptidase. Once again, the clue there being lipids and proteins in the stool. Okay, so let's see why the other ones are incorrect. If it was just protein without fat, then we could look at chymotrypsinogen or pepsin, but that's not the case. We've got lipids and fats. If it was chymotrypsinogen or pepsin, we would have had protein, but really not that fat that we found in the stool. Lipase would have done the opposite. It would have had increased fat in the stool without leading to proteins in the stool. So once again, the fact that we had both is what pointed us to enterokinase. Amylase deficiency will present as sugars or carbohydrates in the stool. So the reason why enterokinase, once again, is the correct answer is because it is important to activate those proenzymes that we mentioned. And if you don't activate those, you will get these proteins and lipids in your stool. So great job, everyone.
Yeah, nice work. Again, a good way to start the evening. Let's head to question number 2. And we did have there's the fact from first aid on pancreatic secretions, so stuff that you can review. We do post the videos of all of our question labs on our YouTube channel, so make sure you go back and review whenever you need. You can always pause on these answers as we go through. Okay, question number 2. Again, we've got those answer choices covered up. We do have a chart for you. And what we're going to do is we'll start with the lead-in. Which of the following organs is most likely responsible for the changes recorded on the graph? Okay, so here we've got an image that you're going to need to refer to, and you've got a good sense of what it is that you're going to be looking for as we dig into the vignette, starting now. A research study used healthy volunteers to determine baseline concentrations of electrolytes in gastrointestinal secretions. Subjects consumed a bolus of food, and investigators measured flow rate, electrolyte concentrations, and pH at multiple sites along the gastrointestinal tract. The data obtained from one collection site were used to construct the figure shown. So again, which of the following organs is most likely responsible for the changes recorded on the graph? Take a look at that. Let us know how many steps you think it's going to take to solve this one as I pass it back over to Sean.
Well, thank you very much, Jeff. So once again, we're going to go ahead and show you what we believe are the important clues in the vignette and lead-in. So we know we're looking for an organ that's going to be responsible for the changes in the graph. And look at the graph, you can see a correlation between what's changing, right? So there's one thing that's relatively constant, it's not changing. There's two things that are constant, and then there's two things that are changing. And if you look at that relationship of how those two things are changing according to each other, that's going to be a huge clue on how to answer this question, okay? So, we're looking at flow rate, electrolyte concentrations, and pH at multiple sites in the GI tract. And that graph is going to show you the data from one collection site. So with the clues I gave you in mind, let's go ahead and take a look at those answer choices. Once again, we'll start at E and work our way up. E, stomach. D, salivary glands. C, pancreas. B, gallbladder. And A, duodenum. So we're going to go ahead and open up that poll and give you a moment to lock in your answers.
Okay, so the poll is open. Go ahead and let us know which organ you think is most likely responsible for the changes recorded on the graph. Nice thing about, making sure that you've got a methodology for attacking these questions is that it really helps you in terms of timing, right? So you kind of you don't have to worry about well, what should I be looking at here, or how should I approach this question? Just you get into this this habit, and every question you can tackle in the same way, reduces some of the cognitive load, and you can really focus on just bashing through these questions. Let's see, we've got about 2/3 of you have voted. I'll give you a couple more seconds and we're going to go ahead and close the poll. If you haven't taken your shot, you could just go ahead, let it rip. You're not being graded here, so take your best shot, and I'm going to close it right now. Okay, let's go ahead and share the results. A little bit tighter this time. Again, answer choice C, pancreas, was the top vote getter, with duodenum trailing. Almost 1/3 of you went with that one. Almost 1 out of 5 of you went with stomach. So interesting breakdown here. So let's go ahead and hide the results. And let's share the answer. And the answer is pancreas. Answer choice C. So again, great work. Almost half of you got that one right. And that's, you know, you guys are off to a great start. If you didn't get it right, that's okay. This is the time to get it wrong. And Sean's going to be here to help make sure you don't get it wrong on test day. Sean?
Well, thank you very much, Jeff. So once again, you know, I gave all of you a clue, and that clue was that relationship between bicarb and chloride, right? And if you look, you can see that the flow rate is increasing, and that's very important in order to answer this question, because what happens is when you have food, and this is probably why some of you guys selected duodenum, right? Secretin is released by the S cells in the duodenum. That then stimulates the exocrine pancreas to increase the flow rate of its secretions, and that will increase bicarb, which will help neutralize the stomach acid contents entering the duodenum. And the bicarb and chloride in pancreatic secretions will have an opposite relationship because they're transported via an antiporter that brings chloride into the cell while sending bicarb out of the cell. Okay, so the correct answer here is pancreas because of that inverse relationship and that increased flow rate. Okay, why are some of the other answers wrong? Well, if it was the stomach or the duodenum, those secretions do not change at varying flow rates. Okay, and you can see here that as the flow rate goes up, those secretions here are in fact changing. Now, if you selected salivary ducts, well, they're going to reabsorb more sodium chloride, which is going to make the solution hypotonic, while the solution is more isotonic at higher flow rates, and we're not seeing that here, right? If you selected the gallbladder, that's going to secrete acidic bile, so there would be a decrease in pH at increasing flow rate. So the key here to answering that question is realizing the release of secretin from the duodenum and the pancreas's response to it, which is going to in turn increase the bicarb concentration and decrease the chloride concentration due to that antiporter. So a tricky question, and I see how some of you selected duodenum, and that was probably the first step, you know, that you were thinking about with the secretin, but you have to take it one step further to realize that as flow rate increases, those concentrations are now reversed. And how does bicarb go up while chloride goes down? Well, that's due to the pancreas responding to the secretin, which was released by the duodenum. So take it one step further, and that's why it's important to think about how many steps the question requires before you answer it. So great job, everyone, and hopefully you'll get it right on test day. Back to you, Jeff.
Yep, that's what's important here. So again, good work, everybody. And we've got the table from first aid on gastrointestinal regulatory substances. So again, just kind of understanding, hey, what are the actions? How does it regulate? What are some of the things that you need to keep in mind? All of that is available for you. So we're about halfway through tonight, and again, we're going to have a raffle at the end of tonight's session, so that somebody will win a subscription to RX 360 Plus. We also have a special that's running. We'll talk a little bit more about it, but you can get up to 55% off a new subscription to USMLE-Rx. But the promo ends tonight. So if you wanted to take advantage of that, especially if you just got a new copy of First Aid or a new copy of either First Aid for Step 1 or First Aid for Step 2 CK, both of those are brand new. If you have the copy of the book or a receipt or just something showing that you've purchased it, you can get that extra bonus in terms of a discount. But we'll talk about that here in a little bit. Let's go to our next question. And again, what we're going to do here is we're going to, we've got those answer choices covered up. We're going to read that lead in. You can see that you've got an image that you're going to need to most likely refer to. So let's go ahead and read the lead in. So which of the displayed changes in protein levels would most likely be found in samples taken 30 minutes after eating compared with blood samples taken prior to eating? Okay. So think about what kind of information you're going to be looking for, and you can look below to see how that information or the answers might be organized as we read the vignette. An endocrinologist is studying the neuroendocrine system responsible for hunger and satiety in physiologically normal humans. To accomplish this, the researcher takes blood samples from healthy volunteers directly before and 30 minutes after a meal. These blood samples are then analyzed for any changes in hormone levels. Which of the displayed changes in protein levels would most likely be found in samples taken 30 minutes after eating compared with blood samples taken prior to eating? Think about how many steps it might take for you to answer this question as I pass it on to Sean.
Well, once again, got a couple steps here, right? We got to figure out what's going on, right? What state is this person in, and what changes are you going to see after somebody ate as opposed to before somebody ate, right? And so that if you look at the next slide here, we kind of show you what we highlighted, but if you really look at that lead-in, it really gives you everything you need to know to answer this question, right? So that lead-in is really telling you everything. So we know now, because we've seen that chart, or the top of it at least, that we're going to be looking at levels of ghrelin, neuropeptide Y, and leptin. So before you look at that chart, anytime you see that, the first thing you want to do is think about, what are those elements? What are they responsible for? When do they go up? When do they go down? And then uncover those answer choices and see what fits that profile, okay? And if you see, and the easiest way to do it is pick the one that you're most comfortable with, right? If you know ghrelin more than neuropeptide or leptin more than the other, look at that one first and rule out the other ones. Then go to the second one and rule out the other ones, and you'll be able to come to that right answer, okay? So with those tips in mind, let's go ahead and uncover the rest of that chart. And as you can see here, we've got a bunch of arrows, right? So we're going to leave it here for a few seconds, let you put your physiology brains to work. So we'll give you a few seconds here just to look at that chart before we open up that poll and allow you to select, hopefully, the correct answer. All right, a couple more seconds here. Well, hopefully that was enough time for you to rule out enough answers to come to the correct one, or pick the correct one right away. So with that, let's go ahead and open up that poll and let you select your answers. We'll give all of you a few moments here.
There you go. Hopefully you already made your selection before we launched it. So go ahead and let it rip. Lots of you do see these types of questions, not just within GI, but the ones where you got to figure out increases, what's staying the same, what's decreasing. So being able to quickly sift and sort these values or what's expected in your mind is a critical skill. So let's go ahead and looks like over almost 3/4 of you have voted. So I'm going to go ahead and close the poll. And we'll share the results. So it looks like you guys have been going with C every night. Everyone's got the C reflex, right? So 44% of you went with that, which is the decrease in ghrelin and increase in neuropeptide Y and leptin. But a little over a third of you went with answer choice D, which was decreases in those first two. So let's go ahead and hide the results and share the answer, and the answer is D. Okay, so tripped up a few of you with this one, but that's okay. If you didn't get this one right, let's break it down so that you are ready the next time you see this. So Sean, can you lead us through this one?
Absolutely. So the reason I'm willing to bet most people got tripped up here is because they forgot the role of neuropeptide Y, right? So what happened here? This patient has eaten, so they're in a well-fed state. So when you're in a well-fed state, what's going to happen? When you're in a well-fed state, you're going to have a couple of changes. Well, somebody who recently ate is going to have low levels of appetite stimulating hormones and elevated levels of appetite suppressing hormones. That makes sense, right? If you've already eaten, we don't want to stimulate your appetite, we want to suppress your appetite. So which ones or one are, stimulating, which ones are appetite suppressing and stimulating? We got to figure that out. So neuropeptide Y is an appetite stimulating neuropeptide, and that's produced by the neurons of the arcuate nucleus upon stimulation with ghrelin. And you can see that here, right? So ghrelin or grelin here is the mnemonic here. So it increases in the fasting state, as you can see here. So what happens here is when you're eating, ghrelin's going to go down because you don't want to stimulate that appetite. So neuropeptide Y and ghrelin are both appetite stimulating. So in the well-fed state, they're both going to go down, okay? Those are both going to go down. Leptin is a major role in suppressing appetite. It plays a major role in suppressing appetite, and it inhibits the release of neuropeptide Y. So what's going to happen here is ghrelin and neuropeptide Y, these are the two proteins responsible for stimulating appetite. They will decrease in a fed state. Leptin is the major hormone responsible for decreasing hunger and appetite, so it will increase in the fed state. So once again, the answer is due to this person being in a well-fed state. So ghrelin and neuropeptide Y, which would stimulate appetite, go down, and leptin is the opposite, and that's going to go up. So that's why the other answer choices are wrong. You simply have to understand what each three of these things are. And like I said, if you get to that point where you're not sure what all three of these do, pick the one that you're most comfortable with, rule out the other answer choices, go to the second one, rule out the other answer choices, and hopefully have one or two left. And at that point, you take your best shot and guess and increase your odds, okay? So just a little test taking tip for you there. Let me hand it back over to you, Jeff.
Excellent, thanks, Sean. And yeah, this is good tips there. We do have a free podcast called the RX Bricks podcast. You can access it from the USMLE-Rx site. All of our episodes are free, and you can download them or stream them through your podcast player of choice. But I linked to the episode on gastrointestinal regulatory substances. So if that's an area in which you'd like to brush up, that's a good one to download. So, and again, we put out a new episode every week, and they're all tied to our bricks. So you can use those for first pass, second pass, or even when you're getting closer to test day, if you need to remediate on a subject, the bricks are a great way to do it. Okay,
Jeff, if you don't mind, we had a nice little mnemonic come through with one of our students here, Sasha. And Sasha tells us the neuropeptide Y makes you hungry, which ends with a Y, and ghrelin is when your stomach is growling. So that's a good way to keep in mind that those two, what those two do. And I saw some comments about people forgetting about neuropeptide Y, so I thought Sasha's mnemonic would be helpful for all of you. So thank you, Sasha.
Yeah, excellent work, Sasha. Good. We put that in the chat so that you guys can copy it down and keep it in your notes. Okay, last question of the night. Again, after this, we're going to do a raffle, so hang tight. Got a pretty short question here, so let's see if we can close strong. We've got all of those answer choices covered up again, and we're going to start with the lead-in. Which of the following salivary concentrations would most likely be decreased in this patient? Okay, so again, you're going to be looking at salivary concentrations and what that patient has been doing that might take it down. So let's read the vignette, quick one. A 25-year-old male comes to the physician with hypersalivation after taking clozapine for 1 month. Physical examination is normal, except for increased saliva production. So again, the question, which of the following salivary concentrations would most likely be decreased in this patient? Let us know in the question box how many steps you think this one will take to solve as I pass it on to Sean.
Excellent. Well, this one is really going to test your physiology. So it is a tough question, right? So which of the following salivary concentrations are going to be decreased in this patient? So in order to get this question right, we need to know, what clozapine does. Well, luckily, they already told us that is causing hypersalivation. So what's going to happen to different concentrations when you have hypersalivation? So luckily, they told us what clozapine does here. If they wanted to be even more tricky, they could have simply mentioned the drug and made you figure out the side effect. But luckily, our test writers were being nice today. So let's go ahead and take a look at those answer choices. E, sodium. D, potassium. C, chloride. B, bicarb, and A, amylase. We'll give all of you a few moments here to lock in your answers.
Okay, poll is a little slow here, but it's up and running. So go ahead and let us know which of the following of these salivary concentrations would most likely be decreased in that 25-year-old male who came to the physician with hypersalivation. Okay, let's see. We've gotten over half of you have voted. This is an interesting one. We've not necessarily an even split, but there's no runaway winner here, at least amongst the choices. So, be interesting to see how this one plays out. Okay, about 3/4 of you have voted, so let's go ahead and close that poll, and we'll share those results. So, I think this was a tricky one here. We've got 25. Yeah, bicarbonate was the leading vote getter, but sodium was only 10 percentage points behind, and that was the lowest vote getter. So I think this one was testing some knowledge that a little bit harder to retrieve. So let's go ahead and hide the results and share the answer. And the answer is answer choice D, potassium. So that was the second place vote getter. It had 22% of the votes for this one. But again, that was almost a completely even spread across all of the answer choices, which means we're going to need to dig into this one. So Sean, let us know how we should have dissected this one.
So, if we're being completely honest here, right, this is one of those questions that's kind of hard to think through. It's doable, but it's one of those things where either you learned this and you have it committed to memory, or you don't. So hopefully after today you will. So let me give you the explanation here, right? We already know that clozapine is causing hypersalivation, so I'm gonna skip through that, right? So saliva, what you need to know, is isotonic at high flow rates and hypotonic at low flow rates, because what happens is the ductal epithelium has more time to modify the secretion by reabsorbing sodium and chloride and secreting potassium, okay? So with increased salivary flow, salivary potassium levels decrease and bicarb also increases. So let's go a little step further, right? So where's saliva secreted from? Well, it's secreted from the ductal acini, and it starts out, as you can see here, isotonic to plasma. Okay, so you can see here that as saliva travels, the ducts and the intercalated ducts reabsorb sodium and chloride and secrete potassium. Okay, and bicarb. So in contrast, at higher flow rates like we have here, saliva has less time in contact with this epithelium. So what's going to happen if you have less time? Well, you're going to reabsorb less sodium and chloride and secrete less potassium. So as a result, the salivary sodium and chloride levels will increase, and the potassium slightly decreases as the flow rate increases. So one thing to keep in mind, and I think the take-home point here is salivary concentrations of amylase, bicarb, chloride, and sodium, they all go in the same direction as salivary flow rate. So if salivary flow rate goes up, amylase, bicarb, chloride, and sodium will also go in the same direction. So if salivary flow rate goes up, okay, amylase, bicarb, chloride, and sodium go up. Potassium is the only one that does not. All right. So keep that in mind. I know it was a tough question, but hopefully you'll commit that to memory today and have an understanding of it and be able to knock this question out of the park on test day.
Excellent. Well, thank you, Sean. And yeah, great work, everybody. This is we tackled some of the some tough concepts here in GI, and you guys did really well tonight. Hopefully, you've learned quite a bit, and you picked up a few tips along the way. We've got the QID numbers here, so you can take a screenshot of this or you jot them down, and then when you are in USMLE-Rx, you can just pop these into Rx search, especially if you want to go a little bit deeper on any of the questions you got wrong or questions that you were a little bit unsure about. Always a good idea to dig into those all the explanations in there, which are really one of the reasons why USMLE-Rx is such a great tool